By the early 1990s, the Florida panther had been reduced to a biological footnote. Fewer than 30 individuals remained in the subtropical swamps west of Miami, scattered across the sawgrass prairies and cypress stands of South Florida. They were not just rare, they were disintegrating. 80% of males born between 1990 and 1992 presented with cryptorchidism, one undescended testicle, or none at all. A strange dermatological marker called a cowlick, a spiral whirl of fur along the spine, appeared in 94% of the surviving population, compared to roughly 9% in genetically healthy puma groups elsewhere in North America. Their sperm counts had collapsed to near-sterile levels. Their hearts carried small septal defects that produced audible murmurs on examination. Their tails kinked at the base in ways no healthy cougar population had ever exhibited. A population viability analysis conducted in 1992 calculated that the Florida panther had a 95% probability of total extinction within 20 years. What happened next became the most consequential genetic rescue ever performed on a large carnivore in the wild, and one of the most instructive conservation experiments of the 20th century. The long slide. The Florida panther did not arrive at the edge of extinction through a single catastrophe. It was pushed there across a century of sustained human pressure. Historically, the subspecies Puma concollicorii ranged across the entire southeastern United States, through Georgia, Alabama, Mississippi, Louisiana, Arkansas, Tennessee, and South Carolina. By the mid-20th century, bounty hunting, habitat destruction, and vehicle collisions had compressed that range into a single remnant pocket in the southernmost tip of Florida. In 1958, the state of Florida finally outlawed killing panthers. But by that point, the damage was so thorough that wildlife officials spent the entire decade of the 1960s unable to confirm whether a single breeding panther still existed anywhere in the state. The species had been bountied, shot from horseback, run over on expanding highway networks, and squeezed out of every corridor that once connected its fragmented populations. In 1972, the World Wildlife Fund contracted a West Texas cougar hunter named Roy McBride to determine whether the Florida panther was already extinct. McBride, who had spent his entire adult life tracking mountain lions through the most remote terrain in the American Southwest, spent months combing the swamps near Lake Okeechobee. He confirmed they were still there, barely. The population was tiny, genetically sealed off from every other puma population on the continent, and already exhibiting early signs of biological collapse. What Melody Roelke found. Through the early 1980s, Florida wildlife officials began capturing the surviving panthers for health assessments. They drew blood, administered vaccines, fitted the animals with radio collars, and tracked their movements through the wetlands. The veterinarian directing much of this fieldwork was Melody Roelke, and what she documented over the following years would ultimately trigger the intervention that saved the species. Roelke had expected to find a small population under environmental stress. What she actually recorded was a biological emergency unfolding in slow motion. The females she examined had cardiac septal defects, small holes between the chambers of the heart that produced murmurs audible through a stethoscope. The males presented with cryptorchidism at rates that defied anything observed in wild puma populations, testicles that had failed to descend properly or had not descended at all, rendering many of the remaining males functionally infertile. Sperm viability among those who could still produce it was devastatingly low. with motility rates and morphology far below the thresholds necessary for reliable reproduction. Parasitic loads overwhelmed their compromised immune systems. Hookworms, ticks, and a host of internal parasites thrived in animals whose immune responses had been weakened by generations of genetic uniformity. Infectious diseases that healthy cougars would survive without difficulty, feline leukemia, feline immunodeficiency virus, were killing Florida panthers routinely. In Roelke's own assessment, the population had reached a biological brick wall. Inbreeding had compounded across generations until every kitten born inherited defect upon defect from both parents. Of the roughly 30 panthers remaining, only 6 females were successfully producing offspring, and those offspring were dying young, being born deformed, or growing up sterile. Roelke sent genetic samples to Stephen O'Brien, a researcher at the National Institutes of Health in Maryland. O'Brien's analysis confirmed what the field data already suggested. The Florida panther exhibited less genetic variation than almost any large mammal population ever studied. They were, in genetic terms, a handful of extended families, cycling the same diminishing pool of alleles through one generation after another. The argument at White Oak. In October 1992, a meeting was convened at the White Oak Plantation Conservation Center in northern Florida. The attendees included geneticists, wildlife biologists, federal representatives from the U.S. Fish and Wildlife Service, state officials from what would later become the Florida Fish and Wildlife Conservation Commission, and National Park Service staff. They had watched the data deteriorate year after year, and they gathered to debate a proposal that had never been attempted at this scale with any large predator. The proposal was called genetic augmentation. The concept was deceptively simple. Capture female cougars from another puma population, transport them to Florida, release them into the Everglades, and let them breed with the surviving males. The resulting offspring would carry fresh genetic material that had not been recycled through decades of inbreeding. The physical defects would, in theory, dilute over subsequent generations. The population would recover its biological viability. The critical question was which donor population to use. The team selected Texas pumas, Puma concolor stanleyana, on the basis that natural gene flow between Florida panthers and Texas cougars had occurred historically, before geographic isolation severed the connection centuries earlier. The Texas cats were not a foreign species being introduced. They were genetic relatives separated by time and geography. The plan provoked fierce opposition. Stuart Pym, a conservation ecology professor at Duke University, later summarized the resistance bluntly. Many people considered it a terrible idea. The core fear was that introducing outside genes would effectively erase the Florida panther as a distinct subspecies. Critics argued that the project would not save the Florida panther. It would replace it with a hybrid and call it the same name. There was also the practical uncertainty. No one had ever conducted a genetic rescue on this scale with a large, free-ranging carnivore. The closest precedents involved smaller species in far more controlled settings: island populations of mice, captive breeding programs for endangered birds, managed herds in fenced reserves. None of those analogues came close to the complexity of releasing 8 wild-caught predators into the vast, uncontrolled wetlands of the Everglades and hoping they would find, tolerate, and mate with animals they had never encountered. The variables were enormous. The Texas females might reject the Florida males. They might fail to establish territories. They might simply die in an unfamiliar landscape. And if anything went wrong, if hybridization produced unexpected problems, if the offspring proved less fit, rather than more, the experiment could not be reversed. There was no recall mechanism for wild cougars released into 1 million acres of swamp. But the alternative, doing nothing, meant watching the subspecies vanish within a single human generation. The transportation costs alone were unbudgeted by any government agency involved until the National Parks Conservation Association stepped in. Will Calloway, their representative on the project, confirmed that his organization agreed to pay for the flights themselves. In January 1995, every federal and state agency involved gave Roy McBride the authorization to proceed. The man who had killed them. Roy McBride was 58 years old when he flew to West Texas to capture the 8 cougars. Born in 1936 in country so remote that reaching the nearest town of Alpine required a 100-mile drive, McBride had spent his entire life in mountain lion territory. The novelist Cormac McCarthy had used him as a source for The Crossing. In a 1991 book about McBride, journalist Donald Schuler captured the strange irony of the man's position. During his younger years, McBride had contributed more to bringing the mountain lion to the edge of extinction in Texas than any other single individual. He had been a professional hunter, killing cougars for ranches and government bounty programs. Now he had been hired to save the subspecies his earlier career had helped to destroy. McBride worked with a blindfolded mule, leading it through the West Texas mountains, flushing cougars from crags and caves. He captured 8 females across the early months of 1995 and prepared them for transport to Florida. TX-104 and the 5 that stayed. The first cougar to arrive was a 4-year-old female designated TX-104. She landed in March 1995 and was placed in a holding enclosure for a brief acclimation period before her planned release. One day after arrival, when handlers opened the enclosure to bring water, TX-104 bolted. She vanished into the Everglades within seconds. No acclimation, no tracking team in position, no controlled release. She survived in the wild for 3 years before being shot dead in 1998 by an unidentified person under unknown circumstances. All 8 Texas cougars were released across the spring and summer of 1995, distributed through Everglades National Park, Big Cypress National Preserve, and adjacent wildlands. 3 of the 8 never contributed to the experiment. One was struck and killed by a vehicle on a Florida highway. Another was shot dead while pregnant, the circumstances unclear. A third died under what wildlife officials described only as somewhat cloudy conditions. That left 5 Texas females loose in the swamps of South Florida, expected to mate with Florida males, carrying generations of accumulated genetic damage. The entire experiment and the future of the subspecies rested on those 5 animals. What the kittens showed. Within 3 years, the results were unmistakable. The 5 surviving Texas females had mated successfully with Florida males, and their offspring were being born without the signature defects that had defined the pre-rescue population. No kinked tails, no cowlicks, no cardiac murmurs. The males had properly descended testicles. Over the following years, those 5 Texas pumas produced at least 20 first-generation hybrid Florida panthers. Each of those offspring went on to mate with other panthers, propagating fresh genetic material through what had been a sealed and deteriorating gene pool. When researchers measured average heterozygosity, the standard metric for genetic diversity, the trajectory was clear. Before the rescue, the Florida panther population sat at 16% heterozygosity. Within a decade, that figure had climbed to 24%. Genetic variation had effectively tripled across the broader population. Kitten survival rates told a sharper story still. Between 2002 and 2004, only 21% of offspring from purely inbred Florida pairings survived to adulthood. Offspring carrying Texas ancestry survived at 53%, more than double the rate. The prevalence of cardiac defects in the hybrid kittens dropped to 8%, half the rate observed in purebred Florida Panthers. By 2003, the last two living Texas Cougars were removed from the Florida wild. They had fulfilled their purpose. Their genetic contribution was already propagating through the next generation without their continued presence. Gold Star Recovery. In 2007, biologists estimated the Florida panther population had crossed 100 individuals for the first time in modern record-keeping. The animals were being documented in territories they had not occupied in decades, moving through ranch lands, pine flatwoods, and hardwood hammocks that their predecessors had been driven out of a generation earlier. Female panthers were establishing home ranges and producing litters in areas where no panther track had been recorded in living memory. In 2010, the journal Science published the formal analysis of the genetic rescue, concluding that panther numbers had increased threefold since 1995. Genetic heterozygosity had doubled. Survival and fitness metrics had improved across nearly every measure, and inbreeding correlates had declined significantly. Paul Bayer, a conservation biologist at Northern Arizona University, described the outcome as a dramatic demonstration of the benefits of genetic restoration. Ron Bass, a retired wildlife biologist at Everglades National Park, put it more plainly. In the history of species recovery, this would rank as one of the gold standard successes, but perhaps the most symbolically powerful moment came in 2017, when a female Florida panther was tracked leading her kittens north of the Caloosahatchee River. It was the first documented crossing of that boundary by a panther with offspring in more than 40 years. The species was no longer merely surviving in one compressed pocket of habitat. It was expanding its range, reclaiming territory lost decades earlier. For a brief window, the 1992 prognosis, the 95% chance of extinction, appeared to have been reversed entirely. The warning underneath. In July 2025, a team of researchers from UCLA and UC Berkeley published a new genomic analysis in the Proceedings of the National Academy of Sciences. They had sequenced the genomes of dozens of post-rescue Florida panthers and compared them against historical samples. The lead author, Diana Aguilar Gomez, addressed the fear that had shadowed the project since its inception, that Texas genes would overwhelm and replace the Florida panther's native genetic identity. The data answered that concern decisively. Modern Florida panthers retained, on average, between 59% and 80% of their original Florida ancestry. The Texas contribution had diluted the inbreeding without erasing the subspecies. The fear of genetic swamping had not materialized. But the same study uncovered a deeper problem. The Texas Cougars, along with an earlier, less documented release of Central American pumas in the 1950s and 1960s, had also introduced harmful mutations into the Florida gene pool. Nearly half of the deleterious mutations now circulating in the modern panther population had originated from these external sources. For the time being, the genetic diversity introduced by the rescue keeps those mutations recessive, masked, unable to express in offspring. But Aguilar Gomez's simulations carried a warning that has alarmed conservation biologists. If the population remains small, around 200 individuals, inbreeding could begin rising again within decades. The masked mutations could start expressing. The defects could return. She suggested that another translocation may eventually be necessary. The threat that isn't genetic. The panther's most immediate crisis, however, has nothing to do with alleles or heterozygosity. In 2024, 36 Florida panthers were recorded dead by state wildlife officials, the highest single-year toll since 2016. The leading cause was vehicle strikes on Florida's expanding road network. In just the first 4 months of 2025, 4 more panthers had already been killed by cars. A proposed 10,000-home residential development sited within core panther habitat is projected to generate enough additional vehicle traffic to functionally eliminate the population if built as designed. Deb Jansen, a retired panther biologist with 3 decades of field experience, expressed her concern without hedging. She feared that people would simply shrug and say, "Well, we tried." 5 cats, 1 lesson. The 1995 Florida Panther Rescue became the foundational case study for genetic augmentation interventions worldwide. Scientists working with isolated populations of mountain pygmy possums in Australia, Swedish adders on fragmented heathlands, and bighorn sheep in California's desert ranges all drew directly from the Florida precedent when designing their own rescue protocols. The experiment demonstrated, with empirical force, that a population teetering on the edge of inbreeding collapse could recover its biological viability through carefully selected genetic infusion. That fresh alleles could mask accumulated defects long enough for natural selection to reassert itself. That 5 mountain lions, captured from the crags of West Texas by a reformed hunter and released into a foreign swamp, could rewrite the trajectory of an entire subspecies. But the same experiment carries a harder truth. Genetic rescue is not a cure. It is a loan against time. It does not buy a future. It buys the opportunity to build one. The Florida panther's continued survival will require habitat corridors, traffic mitigation infrastructure, possible future translocations, and sustained political commitment across generations of decision-makers who never met Roy McBride, who never worked alongside Melody Roelke, and who may never see a wild panther in person. The cats that move through the sawgrass and cypress of South Florida today are the descendants of 26 dying inbred panthers and 5 Texas Cougars that did precisely what they were brought in to do. They are not genetically pure in the way the old taxonomic classifications once tracked. They are something new, a subspecies that was rescued from itself by an experiment most experts expected to fail. Whether that rescue holds depends on what happens in the next 20 years, not the next 200.